Good evening, everyone. My name is Prati Kotari, your host for the day, and it is my pleasure to welcome you all to the 52nd session of Beyond Research, where curiosity meets conversation. Today we are privileged to have with us Mr. Debashish Barik, a PhD scholar and DBT JRF Fellow at the Department of Biophysics, AIIMS Delhi. He is currently working under the mentorship of one of India's most distinguished crystallographer and structural biologist, Professor T.P. Singh. Debashish Sir's research focuses on unraveling the structures of innate immune proteins, a field that holds immense promise for biomedical innovation and drug design. Today, he'll be sharing insights on the topic Decoding Life's Blueprints, the structural power of crystallography. Please join me in warmly welcoming Mr. Debashish Barik, sir.
Good evening, viewers. I am Debashish Barik. I would first like to express my gratitude to Biopectify for inviting me on this talk. So, the topic of my talk is decoding life blueprints, the structural power of crystallography. So, I'm sharing my screen now. Hello, am I audible?
Yes, sir.
Okay. So, first, I would like to uh, introduce to the field of structural biology, and then we will discuss X-ray crystallography. So X-ray crystallography is a subfield of the field structural biology. So structural biology aims to unravel the 3D structure of biomolecules. So basically, we deal with structures in this particular field. So structures uh, like every biomolecules, like proteins and enzymes. So why structures are important to us? As we know, protein has various functions in our body, and in living systems, everything revolves around us. Everything revolves around it. So, solving protein structures are uh, crucial for us. So, understanding structure is key to decoding biological function, mechanism, and disease processes. Various diseases where enzymes are involved in it, and enzyme deficiency diseases are also there. Uh, Many times, protein misfolding diseases are also there. So, every in every disease, proteins are proteins are already there, which is involved in the mechanism of disease progression. So, uh, solving structures of these proteins are crucial, so that we can uh, get into insights the functions and everything around it. So now we will uh, proceed to X-ray protein crystallography. So X-ray protein crystallography is a technique where we uses X-rays to dissect the crystals of protein. So basically, we purify the protein and we form crystals of proteins. Then we used to dissect those crystals to get electron density maps. Then after solving those electron density maps, we used to get the structure. That's how we predict structures of proteins. So it enables insights into active sites and binding pockets and protein ligand interactions and conformational changes. Despite advances in CRAM and NMR, X-ray crystallography remains the most precise method for high resolution structures. Uh, 85% of the structures, protein structures known till now, are solved by XRD, basically X-ray diffraction and crystallography. So, CRAM and NMR in recent advances are also there, but it has limitations to it. So, purpose and scope of the presentation is to explore how crystallography helps decode the molecular blueprints of life, to highlight the impact of protein structure determination on drug discovery and biomedical innovation. So, why crystallography matters today? So, I already told that, as I already discussed, there's that in every disease. there must be a protein involved in it, directly or indirectly. So, understanding protein function at atomic level reveals 3D structure, active sites, and domains. Design precise targeted drugs, enables structure-based drug design via binding site mapping. Visualization of protein ligand and protein inhibitor interactions. So we can visualize protein ligand interactions by solving these structures. Identify disease causing mutations, shows how mutations disrupt protein structure and function, and validate AI predicted protein models, which provides experimental confirmation and refinement of models. And other applications are uncover enzyme and receptor mechanism. We can these models can uh, help to understand enzyme and receptor mechanism and how conformational changes and catalytic reactions happen, and study large protein assemblies. We can also study uh, protein complexes and large protein assemblies and multimeric structures by using X-ray crystallography. And we can develop vaccines and antibody therapies as well. And we can combat drug resistance also, which identifies structural changes in resistance targets by identifying these, and advanced personalized medicines, which links to patient-specific mutations to structural consequences. So, crystallography remains the gold standard for high resolution and structural determination. As I already discussed, that cryo-EM and NMR is also there, but XRD, X-ray crystallography provides high resolution structures, and it is a gold standard method. And uh, all the 85%, over 85% of the structures known till now and deposited in PDB, uh, it contributes to 85% of crystal structures. Basically, XRD contributes to 85% of the crystal structures known till now. And also, it has contributed to nearly 30 Nobel Prizes and revolutionized fields like genetics, immunology, and pharmacology. Even in the age of cryo and AI, like recently AlphaFold, crystallography offers precision, validation, and complementary data crucial for biomedical research. So now we will discuss the crystallography, the history of crystallography, how the uh, progression of crystallography starts and it progresses later on. So the history of recorded protein crystal growth started about 160 years ago. So there is a guy Hinfeld who crystallized first crystallized protein in 1840, which was hemoglobin from the earthworm. This observation clearly stated that protein crystals can be produced by the control evaporation of concentrated protein solution. So basically, by concentrating the protein and by control evaporation, we can crystallize proteins. And protein crystals can be produced by slow dehydration. For the next 15 years, most of the crystals obtained from the blood of several animals were found by chance. The first person to actually devise successful and reproducible methods for the growth of hemoglobin crystals was Funk in 1851. Then the first enzyme which was crystallized was urease, which was crystallized by James Summer in 1926, which was followed by the crystallization of pepsin by John Northrop in 1930. Then these are the history of protein crystallization, how uh, these guys crystallize the protein. But in order to solve the structure, we need the map also. So later on, after discovery of X-ray machine and X-rays, uh, some of the scientists dissect these crystals using XRD, and later on, uh, scientists used to solve these structures and models. So Röntgen discovered X-rays in 1895 while experimenting with cathode rays. His discovery revolutionized medical imaging and later enabled X-ray crystallography. He was awarded the first Nobel Prize in Physics in 1901. So it is the basic traditional X-ray machine which was developed by Röntgen. Then later on, using uh, X-ray machine, Max von Louis dissected the crystals first He was the first who dissected the crystals by X-ray. Then the father-son duo Braggs, W. H. Bragg and W. L. Bragg. Uh, so mm, these father and son duo uh, later on developed Bragg's law, which is uh, used in X-ray diffraction. So the two physicists, Bragg and his father Henry Bragg, in 1912 and 1913, developed Bragg's law, which connects the scattering with evenly spaced planes within a crystal. The Bragg's father and son shared the Nobel Prize in 1915 in physics for their work in crystallography. So they gave Bragg's law, which used in XRD to solve the Miller indices as well atomic model also. So later on, after development of all these, after some decades, James Watson and Francis Crick gave the double helical structure of DNA by solving the diffraction data of Rosalind Franklin. Basically, Rosalind Franklin diffracted the DNA. Then, by using uh, her their data, Watson and Crick proposed the double helix structure of the DNA. Then many of the groups started working on structural biology and XRD and protein crystallography. Then the latest developments are CRIAM, AlphaFold, and several other AI tools. Recently, in 2020, AlphaFold, uh, some of the groups in Alphabet, which is a subsidiary of Google, they build AlphaFold, and they later on they got Nobel Prize in 2024. Uh, these are the scientists, Demis Hassabis and John Jumper, they and David Becker. They got Nobel Prize in 2024 in chemistry for their work in computational protein design due to development of AlphaFold. So these are some structures and Nobel Prizes given in the field of crystallography. So as we already discussed, Max von Lee was the first to diffract the X-ray crystals using X-rays, uh, protein crystals using X-rays, and Perudes and Kendrew got their Nobel Prize in chemistry for their studies of structures of globular protein in early 60s. Then in 1962, James Dewey Watson, uh, Crick, and Wilkins got the Nobel Prize for solving the structure of DNA by using XRD data of Rosalind Franklin, which I already discussed. In 1964, Dorothy Hoskins got the Nobel Prize in chemistry for XRD determination of structure using XRD, the structures of important biochemical substances. Then in 1972, Stanford Murray, William Epstein got the Nobel Prize in chemistry for understanding the connection between chemical structure and catalytic activity of the active center of ribonuclease molecule, which is an enzyme. And then later on in 1976, William N. Lipscomb got the Nobel Prize in chemistry for his studies on the structure of boranes, eliminating problems of chemical bonding. Then in 1885, Jerome Carle, Herbert A. Hauptmann got the Nobel Prize in chemistry for the outstanding achievements in developing direct methods for the determination of crystal structures. Then later on in 1988, this group of Diesenhofer, Michael, and Robert Hoover got the Nobel Prize in chemistry for the determination of 3D structure of photosynthetic reaction center. Then in 1997, John E. Walker got the Nobel Prize for his work in understanding the enzymatic mechanism of the synthesis of ATP. Then in early 2000, in 2003, Roderick Mackinnon, Peter Avey got the Nobel Prize in chemistry for their work in structure of aquaporins, the water channels in cell membranes. Then in 2006, Roger Kornberg got the Nobel Prize in chemistry for his studies of the molecular basis of eukaryotic transcription. Then in 2009, Adai Unit, Thomas A. Seeds, and Venkataraman Ramakrishnan got the Nobel Prize for solving the structure of ribosome. Venkata Ramakrishnan, or as you know, he is Indian by origin. And in 2012, Brian Kovilka got the Nobel Prize in chemistry for his studies in G-coupled receptors. Then, as we know, recently in 2024, the group got the Nobel Prize in chemistry for their development of AlphaFold and their contribution in computational uh, design of this AI tool. How protein structures are determined. Now we will understand the step-by-step process. First, we used to uh, uh, clone our target protein. Then we used to express the protein in expression system. Then we used to purify these proteins. After purification, it is followed by crystallization. We put the plates uh, for crystallization. Then we used to check the plates after some time for crystals. Then after getting crystals, we will check whether the crystals are good enough to diffract. Then we used to diffract the crystals either in-house facility, X-ray facility, or in synchrotron facility. Then we used to collect the diffracted data. Then we proceed for structure determination. Then after solving the structure, we used to carry out validation analysis and other bioinformatics uh, uh, studies. So now we will proceed to our next slide. So protein to crystals. Now we will discuss the whole procedure of crystallization, how we get crystals from proteins. So in order to understand crystallization, we will go through some aspects of the crystallization. First, we need the protein sample must be uh, highly pure. So we need high purity of protein sample, requires highly purified monodispersed protein, and concentration of the protein should be 10 to 40 mg per ml, depending upon the protein and its activity, basically its function. So uh, next, the precipitant screening. Which precipitant is likely to help to develop to form the crystals? So choice of precipitant is crucial for development of for uh, forming of for growth of crystals. So we need to play around these precipitants. So we can use screens for this. We can carry out different kind of buffers and precipitating agents of different concentrations, and we can optimize our crystallization process. Buffer and pH optimization. Buffer, we can, as I already told, choice of buffer is also necessary. So we can try a different kind of buffers, depending upon its the PI of protein and also the stability. So pH affect, as we know, pH affects protein charge and crystallization behavior. So choice of buffer as well as choice of precipitant is very crucial for the optimization step or, or for growth of crystals. And then temperature control. Crystallization is typically done at 4 degrees Celsius to or 20 degrees Celsius, depending upon the protein stability and the type of protein. So we need to optimize uh, everything according to from protein to protein. It varies from protein to protein. Then slow supersaturation. All methods aim to slowly bring the protein into supersaturated state where nucleation and crystal growth can occur, achieved through vapor diffusion method, oil overlay or LCB matrix diffusion. We will discuss all these crystallization methods in the upcoming slides. Trial setup in small volume. As I already told, we need to play around our crystallization process. So we should use nanoliter to microliter volumes for high throughput condition screening, and robotic or manual steps can be often employed depending upon the laboratory setup, whether a robotic facility is there for crystallization or we need to manually crystallize. So next is use of crystallization screens. As I already told, we can use uh, commercial screens as well, uh, where there must be different kind of buffers and different kind of precipitants with different concentrations. So we will put our protein for crystallization in these screens with varying concentrations, varying precipitating agent, and varying buffers. So in order to get crystals, good crystals. So next process, next step is visual monitoring. After plating the protein for crystals, we need to check after days, after weeks, so whether the crystals are growing or not. So whether it is growing or not, or whether it is evaporating or not. So crystallization drops are monitored under a microscope regularly. It can take up to hours, up to days, up to weeks, or maybe up to years, depending upon the protein and protein yield. And observation of nucleation, precipitation, or crystal growth informs optimization. Now we will proceed to our next slide. Now we will discuss protein crystallization. So this is the phase diagram in the left side. Uh, in the y-axis there is protein concentration, and in the x-axis there is precipitant condition. So depending upon the protein concentration and precipitant concentration, these are the solubility curve, and some zones uh, restricted to this phase diagram. We will discuss every zone, each and every zone. So under saturation zone uh, is the zone where protein is fully soluble, and as the name suggests, under saturation, it is not saturated. So basically, there is no nucleation or growth happens in this region. So crystals will not form in under saturation zone. Then there is metastable zone. Crystals can grow if nuclei are already present, but nucleation won't start spontaneously. If there is already crystals are already grown. If we put that grown crystal in this particular zone, metastable zone, then only it will grow. Otherwise, the protein will not grow in this particular zone, metastable zone. Then nucleation zone. This is the zone where actually crystal nucleus forms. So actual growth of crystals occur in this particular region, which is nucleation zone. So supersaturation is high enough for spontaneous formation of crystal nuclei. Once the crystal is formed in nucleation zone, if we put those crystals in metastable zone, if there will be metastable condition of metastable zone, then these crystals will able to grow. But nucleation occurs only in nucleation zone. Then there is precipitation zone. If there is excess supersaturation, it can cause amorphous aggregates or non-specific precipitation, which will not help to get our crystals. So basically, crystals will not grow in this particular region. Then some important points to focus on. Crystallization is most effective when nucleation occurs in the nucleation zone, as I already discussed, followed by growth in the metastable zone. Fine control over the shift between these zones is achieved by varying pH, temperature, salt, tags, and protein concentration. So we can shift these regions by playing around with pH, temperature, salt, and precipitating agent, and buffers and protein concentration as well. So the next is supersaturation, which is the driving force, and precise control is key for obtaining well-ordered crystals. Seeding techniques can shift crystals from nucleation to growth in metastable conditions. We can uh, uh, take small crystals and put it for crystallization. So it will be in metastable zone, and metastable zone will help this nucleus or nuclear crystals to grow, which will fasten the process of crystallization. Then time is also a factor for crystal growth. As I already told, it can take up to hours, up to weeks, or maybe up to years, depending upon the protein. So next we will discuss the crystallization techniques. There are several techniques. to uh, put our protein for for crystallization. So first is batch crystallization, the second is dialysis, the third is vapor diffusion, and the next one is free interface diffusion method. So we will discuss these one by one. So batch crystallization, protein and precipitant are mixed together in a single step, and crystallization occurs as the solution reaches supersaturation. So it is a very traditional method. Then dialysis, protein solution is enclosed in a semi-permeable membrane and dialyzed against the precipitant solution, allowing gradual exchange and control supersaturation. The next is vapor diffusion method. It is a widely accepted and followed method for crystallization, and there are two methods: hanging drop vapor diffusion method and sitting drop vapor diffusion method. So in hanging drop diffusion method, a drop containing protein and precipitant is placed on the underside of a cover slip. First, we will put a buffer uh, in the plates in the reservoir. Then we will put our protein and in the cover slip with precipitant. Uh, After mixing with the precipitant in the cover slip, then we uh, put the cover slip upside down uh, on that plate and on that well after applying silicone, so that there will be no air exchange of air. So water vapor diffuses from the drop to the reservoir, uh, which is located below, and concentrating the drop and promoting crystallization. So basically, uh, all the extra uh, water vaporizes and it it goes to the reservoir. So this is our hanging drop vapor diffusion method. Next is sitting drop vapor diffusion method, where we uh, the protein precipitant mixture we used to sit in a pedestal, present pedestal just beside the reservoir chamber. So there is a platform. We used to put our protein and precipitant mixture in the platform, and we allow it for vapor diffusion, and which drives the supersaturation and crystal growth. So these are the two most widely accepted or widely used. This mainly we used hang drop vapor diffusion method, but sitting drop is also people used to do. The next is free interface diffusion method. Free interface diffusion method, protein and precipitant solutions are layered or flow into contact, allowing slow diffusion and gradient formation for controlled crystallization. Now we will move into our next slide. So why we use X-ray for crystallography and not any other rays in the electromagnetic spectrum? So uh, we can, uh, as you can see this table, light has a wavelength of 300 nanometer, and individual we can visualize individual cells and subcellular organelles uh, through the light microscopy. By using electron microscopy, which has a wavelength of 10 nanometer, we can get insight into the cellular architecture and shapes of large protein molecules. But X-rays has a wavelength of zero point 0.1 nanometer, which is 1 angstrom, so which helps to, which is in order of the atoms, so we can get the atomic detail of protein. That's why we use X-rays for the diffraction of crystals, and then we proceed to solve the structure of proteins in order to get the atomic detail. So as I already told, X-rays is in the order of atom diameter and bond length, allowing these to be individually resolved. No lenses available to focus X-rays, so we don't require any lenses. Crystals acts as a magnifier of the scattering of X-rays. So when these X-rays scatter the crystal act as a magnifier. So firstly, we will discuss how we get crystals from protein. So once we get crystals, we will get our structure from crystals. So uh, as the name of the slide suggests, name of the slide is crystals to structure. We will discuss how we get the crystals, we get the structure from these crystals by solving the XRD data. So for diffraction, we need to assure whether the crystals formed are good enough to diffract or good enough to get the atomic detail. So for that, crystal must be small in size, around 1 millimeter. It should be perfect. There must be no cracks, no inclusions such as air bubbles. And these are the steps shown in the right side. So steps after getting crystals, we will first diffract the crystals in a synchrotron facility or in-house X-ray facility. Then we will collect the data. After data collection, we will work on its phase problem. We will fix the we will add the phase to our X-ray data. Then we will get the electron density. Then after getting the electron density, we will uh, solve the structure and get into the atomic details. Then we will build the model. Then after model building, we will refine the structure. Then we will get our final model. And after getting the final model, we can also validate those things with using bioinformatic tools, as well as we can carry out validation assays, which can be biophysical or biochemical, depending upon the function and protein. So we will move to next slide. Data collection. So how data collection goes on. So first we need to mount the crystals. So how we will discuss how we will mount the crystals. Crystals are mounted in a way so that the sample can be rotated and an X-ray beam can be passed through the sample. So we put the sample in the XRD goniometer. Then it it the goniometer used to rotate, and it it basically uh, so it basically uh, put the X-ray beam. Uh, so X-ray beam passed through the sample. Then it gave the diffraction pattern in a film or in an electronic device. So after getting that electronic electron uh, after getting that those data was recorded in an electronic device. After recording that those data, we used to collect those data for further analysis to fix the phase and then uh, building of atomic model. Then methods of mounting include using either a capillary or a tube. Both capillary and tubes are mounted on a goniometer, as I told. So exposing X-rays, once the crystals are correctly mounted, they are exposed to X beams. As I already discussed, X-ray sources include a synchrotron. Synchrotron is a big facility for X-ray diffraction, and it can give a high resolution structure, nearly 2 angstrom or so. But in-house facility, in-house X-ray diffraction facility, we, we will not get high resolution. So synchrotron facility help in getting high resolution data. So the source of the X-rays is often a synchrotron. The typical size of a crystal for data collection may be zero point 0.3, and zero point 0.1 millimeter, which is a these are the dimensions for the crystals. The crystals are bombarded with X-rays, which are scattered from the planes of the crystal lattice. The scattered X-rays are captured as a diffraction pattern on a detector, such as film or an electronic device. Rotate crystal through 1 degree and record XRD pattern. So the crystal was rotated one by 1 degree, uh, and every pattern was recorded varying uh, 1 degree. So if XRD pattern is very crowded, we used to reduce the degree of rotation and repeat until 30 degrees were obtained. sometimes 180 degrees depending on crystal symmetry. So lower the symmetry, more data are required. For high resolution, we use synchrotron as I already discussed. So after diffracting those crystals, we used to get the diffraction pattern, and these diffraction pattern was recorded in a diffraction in an electronic device. So this is the diffraction pattern from a protein crystal. Uh, these are the dots you can see over here. So after getting this diffraction pattern, we used to get data. After solving data from those data, we only used to get amplitude of the after the diffraction. We only used to get amplitude of the uh, atoms, but we need to, in order to solve the structure and build atomic model, we need a phase also, so that uh, we need to uh, add phase over amplitude uh, in order to get the these kind of density, which is shown over here in the right side. The 3D structure obtained above is the electron density map of the crystal. This kind of density is there where we used to uh, build our atomic model of the protein. In every density, there will be a chain of atoms of proteins. So this is the density. This density suggests is something is there which was diffracted. So something is there. It must be an atom of protein only. So by using this density map, we used to obtain our atomic model of the protein. So as I already told, we used to only get amplitude of from the diffracted data. In the measurement of data from an X-ray crystallography experiment only, the amplitude of the wave is determined. To compute the structure, the phase must also be known. So how we will get the phase? So what we used to do, we take a homologous structure similar to our protein of interest, which has high sequence similarity. So we used to take that homologous structure. We take the phase of that homologous structure, and then we put that phase in our amplitude of the protein of interest. So basically, we uh, sum up the phase of the homologous protein and the amplitude of the protein of our interest. So after uh, adding the phase of homologous protein into our protein of interest, we used to get the electron density map, which is here in this slide. So after getting this kind of electron density map, we used to build our atomic model later on using various tools. So since it cannot be determined directly, it must be determined indirectly or by some other experiment. So this line suggests that we don't have any phase after diffraction. We don't have any phase of our own protein. So what we used to do, we uh, take the phase of homologous protein, which has a high sequence similarity with our protein of interest. But we have only amplitude of our own protein. So what we used to do, we add our amplitude with the phase of the homologous protein, and we used to get the electron density map. So this slide represent how the phasing we used to carry out. So these are the multiple methods which focus on the phase problem. So molecular replacement is there, multiple or single isomorphous replacement is there, and multiple single wavelength anomalous diffraction is there. So uh, all these methods of solving the phase problem includes this principle of Fourier transform. So next slide is structure determination. So after fixing our phase, first we got our amplitude from the diffraction data, amplitude of protein of our interest. Then we fix the phase because we don't have any phase for our protein. So what we used to do, we take the phase from another homologous protein. Then after fixing the phase, we used to get the electron density map. Then once we used to get our electron density map, what we used to do, we used to predict the structure. We used to determine the structure. So this is the next step, structure determination. So fitting of protein sequence in the electron density, it involves fitting of the protein sequence in the electron density. So as you can see, there is a density over here in blue color, which which implies something is there, some atomic chains of proteins are there, which we need to fix. So whatever space is available, we used to build our atomic model in that space only, the designated space only. So electron density is not self-explanatory, can be automated if resolution is close to 2 angstrom. So uh, as I told, to build a better atomic model, resolution, good resolution should be there. So data with 2 angstrom or close to 2 angstrom is better to build a precise atomic model, and it can be interpreted is largely defined by resolution. As I already told, atomic model and structure determination of proteins is highly dependent on the resolution of protein. So once we used to build our or fix the uh, atomic chains, the protein chains in these electron density map, we used to carry out refinement using software such as RefMac. So these softwares like RefMac refine the atomic model solved by the solved by us after getting the electron density map. So after building the atomic model, we used to carry refinement. After refinement, uh, we used to carry out validation from PDB or from PDB. So validation report uh, suggests validation report has various parameters which suggest whether the atomic model built by us is precise enough or not, or whether it is doubtful or what, or else we need to, if it is doubtful or the values are not good, we need to fix this atomic model. So the next slide is summary of X-ray crystallography. So as I told you, X-ray crystallography uh, is X-ray crystallography is a good method to determine structures. And although after recent advancements like NMR and as well as CRIAM, these uh, advanced techniques don't provide high resolution data uh, like XRD, but they have other roles also where XRD used to fail. But most of the structures can be solved by X-ray crystallography. So we will discuss advantages and disadvantages of X-ray crystallography. Advantages, it is very routine and high potential for automation. Uh, Fast structure determination can be carried out using X-ray crystallography. Once we get data, we can solve the data in hours, in some hours, in order to uh, determine the structure. And disadvantages it has, such as crystal growth is a major unsolved problem. If the crystal, if uh, there will be no crystals after months or so, we need to carry out our whole crystallization process once again. So crystal growth is a major problem. Once crystals may be obtained at irrelevant conditions, this is also a problem. Sometimes some ligand is also there. density of a ligand is also there, which we need to fix, and other heterogeneity is also there, which we need to fix. So then there are multiple irrelevant conditions in crystals. Crystal packing forces may influence structure, and aqueous protein exists in conformational ensemble. It can be argued that X-ray may filter for the conformation that crystallizes. So whatever conformation it has during the crystallization process, during it forms the crystals, it basically the XRD only diffract only that conformation only. And the next is glycosylation, will normally render the protein hard to crystallize. So proteins with glycosylation uh, is very tough to crystallize. Multi-unit proteins are also hard to crystallize. So these are the disadvantages with this. We will now move to our next slide. Why structure matters. Why we need to solve these structures. As I already told, solving protein structures can help us to understand various diseases and progression of diseases, and various underlying mechanisms involved in the disease progression, and various mechanism also which is going in our body, which are not known. So relation it also uh, provide insights into relationship between protein structure and function. The 3D structure of protein determines its biological activity, stability, and interactions in living systems. Changes in structure by mutation or misfolding, as there are multiple mutational diseases or misfolding diseases. So, how these structure, protein structure changes abruptly in mutation. So, where which gene usually mutate in a particular disease, so that it contributes to change in a conformational changes in a particular site of a protein. Uh, Changes in structure can disrupt function or cause disease. Example: hemoglobin quaternary structure allows oxygen binding and release in tissues. Examples in biology and disease, we can discuss two protein misfolding diseases here, such as in Alzheimer disease. There is a misfolded amyloid beta amyloid protein. So understanding the structure of this particular protein can help to cure Alzheimer disease in the future. Structural mutation in cystic fibrosis protein can also help to understand the underlying mechanism of cystic fibrosis. Structure-based drug design. Crystallography guides drug development by revealing binding pockets and active sites. So it also reveals the binding pockets and active sites in a protein or in a enzyme. So, example in HIV protease inhibitors. HIV protease inhibitors were designed using its crystal structure, and imatinib targets the BCR-ABL fusion protein in chronic myeloid leukemia. So, understanding structures can help in understanding various mechanisms uh, related to diseases and its progression. Now we will move to next slide. What are the challenges in structural biology which we need to work on? So, crystallization difficulties are there, dynamic and disordered proteins are there, and uh, when how how we use and when to use KM and AI when we when the XRD fails to predict the structure. So crystallization difficulties. Obtaining well-ordered protein crystal is often the rate limiting step. As I already told, we need to during crystallization we need to play around the precipitating agents as well as buffers and their concentrations. Membrane proteins, large complexes and flexible regions are especially challenging. So membrane proteins are very challenging to crystallize and to build their atomic model to solve the structure. And crystallization process requires extensive screening of buffers, pH, precipitant, and additives. Even successful crystals, even after getting successful crystals, it might it might not reflect well or provide high resolution. If we will not get high resolution, we will not be able to uh, get the electron density map in order to solve the structure. And the next is dynamic and disordered proteins. Many proteins have intrinsically disordered regions that don't crystallize. So one need to work on that. Flexible domains may be essential for function but are invisible in crystal structures. So uh, flexible domains are essential for function, but we can't differentiate the flexible domains in crystal structures. And X-ray crystallography captures static snapshots. As we know, whatever conformation it would be, once the crystallization occurs, it was static. So we will only get the static conformation of the protein, and it limits insights into dynamic processes like allostery and folding pathways. So it doesn't provide any dynamic uh, information. Now we will discuss CRIAM and AI. So CRIAM excels at imaging large flexible complexes without needing crystals. So CRIAM is only helpful when we need to take and the images of large protein complexes, which is hard to get the data from by diffracting from XRD. AlphaFold predicts the protein structures from sequence with remarkable accuracy. Whenever we can't get crystals, we can use AlphaFold to predict the structure. Sometimes what happens is the crystals may diffract or not may diffract, but some of the regions don't provide any information. For those reasons, we can use AlphaFold to predict the structure of only that section. Both tools complement crystallography, as I already told. Whenever crystallography fails, it complements to the crystallography, especially uh, for hard to crystallize targets. Those targets which are hard to crystallize, we can use CRIAM and AI, such as AlphaFold, for those proteins or for those particular regions where we don't get any information from that electron density map. So crystallography remains a vital uh, method for validation, refinement, and drug level resolution. Now we will move into our next slide, which is future outlook, emerging trends, and innovation. So integration with artificial intelligence. So we need to integrate crystallography with AI so that we can uh, solve our atomic model uh, in a better way. So tools like AlphaFold and Rose TTA Fold are revolutionizing structure prediction. X-ray crystallography remains essential to validate, refine, and complement predictive structures. AI can assist in crystal screening, model building, and automated refinement. During crystallization, we need to uh, select our screens and precipitant. So AI can also assist in that that so that we can use our screens in a better way. What screens should we use, or what precipitant should we use in order to get appropriate crystals. Crystallography beyond static structures shifts towards capturing dynamic conformational state. As what I already told, crystallography only provide static snapshots, static electron density data. So, uh, moving into time-resolved crystallography, which is a recent advancement. So, time-resolved crystallography can provides uh, dynamic conformational states. It helps to visualize enzyme mechanism, protein folding, and ligand-induced changes in real time. So it might help in understanding various protein folding diseases such as Alzheimer's and Parkinson disease and cystic fibrosis as well, etc. So next is miniaturization and automation. So high throughput crystallization and robotic pipelines for rapid screening. We can use robots for high throughput crystallization and robotic pipelines, integration with microfluidics and machine learning to enhance success rates. If we uh, uh, if we convert the whole process into automation and we if you use robots and machine learning, we can automate our whole process of crystallization and the which will help to enhance X-ray crystallography. So the next is crystallography for complexes and membrane proteins. As I already told, crystallography, it is hard to solve complexes, structure of complexes, hard to get that affected the structure of crystals of complexes and membrane proteins. And membrane proteins are very tough to get. It is very tough to get crystals of membrane proteins. So ongoing advances in crystallization techniques enable solving challenging targets critical for drug design, viral proteins, and GPCRs. Now we will move to our next slide. Structural biology as a collaborative ecosystem and clinical and translational expansion. So why we should build a collaborative ecosystem in structural biology? So we should build a collaborative ecosystem in order to solve the structures as many of the instances XRD X-ray crystallography fails to predict the structure, as I already told you. In once some proteins we used to get the diffracted data, but some sections are missing, and some sections, some electron density maps are don't provide any specific detail. So, and for those sections, we can use other tools such as NMR or AlphaFold or computational modeling. So, crystallography will continue to play a central integrated role in this toolkit. So, crystallography will play a major role, but all other techniques will help to aid the crystallography. Next is clinical and translational expansion. Accelerating structures guided drug design for cancer, infection, and genetic diseases. Personalized medicine through structure, function, and mutational analysis. Now we will move to our next slide. So we will like to end here. So the take-home message is here is that structure without function is a corpse, and function without structure is a ghost. As, as I told you, structure is everything. Without structure, we will won't be able to get insight into the function of the protein and underlying mechanism involving with the disease and disease progression. So I would, this is the acknowledgement page. I would like to express my gratitude to Biophysics, and I would like to express my gratitude to my supervisor, co-supervisor for the valuable guidance in my PhD work, and I would like to express my gratitude to AIIMS Delhi as well as my family and friends. So thank you viewers for enjoying this session.
Thank you so much, sir, for such an insightful talk. Participants, if you have any doubts, you can ask sir right now.
Yeah, any questions please?
And I hope the mic and video are accessible to you as well. And also, you can type your questions in the chat box.
Viewers, you may ask questions or doubts.
Participants, if you have any doubts, you can answer it now. We still have a few more minutes before we conclude the session.
If any of you have any questions, you can ask. You can later on connect to me on LinkedIn as well if you have any doubts.
I think there are no more doubts. Can we conclude the session?
Yes, yeah, of course.
So, thank you so much everyone for joining us today. And I have also shared the feedback form link in the chat box. So, do make sure to fill that for enabling your e-certificates for this session. And thank you so much, sir, for such an insightful talk and uh, like, uh, engaging discussion. Thank you so much, sir, and thank you all for joining us today.
Thanks, many thanks, and all the best to all the viewers.
Thank you so much, sir.